तो इसके बाद आता है मॉलिक्यूलर बेसिस ऑफ द इनहेरिटेंस, जिसमें हम देखेंगे डीएनए के बारे में, द स्ट्रक्चर ऑफ द फॉर द जेनेटिक मटेरियल, आरएनए वर्ल्ड देखेंगे, रेप्लिकेशन देखेंगे, ट्रांसक्रिप्शन देखेंगे, जेनेटिक कोड देखेंगे, ट्रांसलेशन देखेंगे, रेगुलेशन ऑफ द जीन एक्सप्रेशन देखेंगे, ह्यूमन जीनोम प्रोजेक्ट और डीएनए फिंगर प्रिंटिंग देखेंगे। तो एनसीईआरटी कहता है कि इन द प्रीवियस चैप्टर यू हैव लर्नड द इनहेरिटेंस पैटर्न एंड द जेनेटिक बेसिस ऑफ सच पैटर्न। एट द टाइम ऑफ द मेंडल द नेचर ऑफ दोज फैक्टर रेगुलेटिंग द पैटर्न ऑफ द इनहेरिटेंस वाज नॉट क्लियर। Over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA, deoxyribonucleic acid, is the genetic material at least for the majority of the organism. In the class 11, you have learned that the nucleic acid are the polymers of the nucleotides. Deoxyribonucleic acid (DNA) and the ribonucleic acid (RNA) are the two types of the nucleic acid found in the living system. DNA acts as the genetic material in most of the organism. RNA, though it also acts as genetic material in some viruses, mostly function as messenger. RNA has additional roles as well. It functions as the adapter, structural, and in some cases as a catalytic molecule. In class 11, you have already learned the structure of the nucleotides and the way these monomers units are linked to form the nucleic acid polymers. In this chapter, we are going to discuss the structure of the DNA, its replication, the process of making RNA from the DNA transcription, the genetic code that determines the sequence of the amino acid in the proteins, the process of the protein synthesis translation, and the elementary basis of their regulation. The determination of the complete nucleotide sequence of the human genome during the last decade has set a new era of the genomics. In the last section, the essentials of the human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In the subsequent section, we will understand that why it is the most abundant genetic material and what its relationship is with the RNA. So the DNA is a long polymer of the deoxyribonucleotides. The length of the DNA is usually defined as the number of the nucleotides for a pair of the nucleotide, referred to as the base pair present in it. This alpha is the characteristic of an organism. For example, a bacteriophage known as the 5 into 124 has 5,386 nucleotides. Bacteriophage lambda has 48,502 base pair. Escherichia coli has 4.6 into 10 to the power 6 base pair, and the haploid content of the human DNA is 3.3 into 10 to the power 9 base pair. Let us discuss the structure of such a long polymer. First, the structure of the polynucleotide chain. So let us recapitulate the chemical structure of a polynucleotide chain, DNA or the RNA. A nucleotide has three components: nitrogenous base, a pentose sugar, ribose in the case of the RNA, and deoxyribose for the DNA, and a phosphate group. There are two types of the nitrogenous bases: purine, cytidine, and guanine, and the pyrimidine, cytosine, uracil, and the thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil present in the RNA at the place of the thymine. A nitrogenous base is linked to the OH of the 1-C of the pentose sugar through the N-glycosidic linkage to form a nucleoside, such an adenosine or a deoxyadenosine, guanosine or a deoxyguanosine, cytidine or the deoxycytidine, and the uridine or the deoxythymidine. When the phosphate group is linked to the OH of the 5-C of a nucleoside through the phosphate ester linkage, a corresponding nucleotide or a deoxynucleotide, depending upon the type of the sugar present, is formed. Two nucleotides are linked to the 3-5-phosphate ester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free uh, a free phosphate moiety at the 5% of the sugar, which is referred to as the 5% of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of the 3-C sugar, which is referred to as the 3' end of the polynucleotide chain. The backbone of the polynucleotide chain is formed due to the sugar and the phosphate nitrogenous base linked to the sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. Also in the RNA, the uracil is found at the place of the thymine 5-methyl-uracil, another chemical name of the thymine. DNA as an acidic substance present in the nucleus was first identified by the Friedrich Misser in the 1859. He named it as the nuclein. However, due to the technical limitations in isolating such a long polymer intact, the elucidation of the structure of the DNA remained elusive for a very long period of time. It was only in the 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by the Maurice Wilkins and the Rosalind Franklin, proposed a very simple but famous double helix model for the structure of the DNA. One of the hallmarks of their proposition was the base pairing between the two strands of the polynuclear chain. However, this proposition was also based on the observation of the Irving Chargaff that for a double-stranded DNA, the ratio between the adenine and the thymine and the guanine and the cytosine are constant and equals 1. फिर कहता है the base pairing confers very unique property of the polynuclear chain. They are said to be complementary to each other, and therefore if the sequence of the bases in the one strand is known, then the sequence in another strand can be predicted. Also, if each strand of the from the DNA, let us call it as the parental DNA, act as a template for the synthesis of a new strand, the two of double-stranded DNA, let us call them as the daughter DNA, thus produced would be identical to the parental DNA molecule. Because of this, the genetic implication of the structure of a DNA become very clear. फिर आता है the silent features of the double helix structure of a DNA are as follow. It is made of the two polynuclear chain where the backbone is constituted by the sugar phosphate and the bases project inside. दूसरा, the two chains have the anti-parallel polarity. It means if one chain has the polarity 5' to 3', the other has the 3' to 5'. The bases in the two strands for the pair through the hydrogen bond, H bonds forming base pairs. Adenine forms two hydrogen bonds with the thymine from the opposite strand and vice versa. Similarly, guanine is bonded with the cytosine with the three hydrogen bonds. As a result, always a purine comes opposite to the pyrimidine. This generates the approximately uniform distance between the two strands of the helix. Figure that the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer. A nanometer is 1 billionth of a meter, that is 10 to 1 nanometer, and are roughly 10 base pair in each turn. Consequently, the distance between the base pair in the helix is approximately 0.34 nanometer. The planes of the one base pair stacks over the other in a double helix. This, in addition to the H-bond, confers stability to the helical structure. Compare the structure of the purines and the pyrimidines. Can you find out why the difference between the two polynucleotide chain in the DNA remains almost constant? The proposition of a double helix structure for the DNA and its simplicity in the explaining the genetic implication become revolutionary. Very soon, Francis Crick proposed the central dogma in the molecular biology, which states that the genetic information flows from the DNA से RNA से protein. फिर कहता है कि in some viruses the flow of information is in the reverse direction, that is from the RNA to DNA. Can you suggest a simple name to this process? जिसको हम reverse transcription कहते हैं. तो डीएनए का डीएनए कॉपी बनाना रेप्लिकेशन हो गया. फिर डीएनए से एमआरएनए बनाना ट्रांसक्रिप्शन, और एमआरएनए से प्रोटीन बनाना ट्रांसलेशन. इसी को हम सेंट्रल डॉगमा कहते हैं. फिर ये डीएनए का डबल हेलिक्स मॉडल दिया हुआ है. एडनिन ए टी है जो एक टी के साथ पेयर करता है, और जी है जो सी के साथ पेयर करता है. फिर डबल स्ट्रैंडेड पॉलीन्यूक्लियर चेन दिए हुए. इधर हो गया 5', इधर हो गया 3', और फिर इधर हो गया 5', तो इधर हो गया 3'. और यहाँ पर ए और टी के बीच 2 हाइड्रोजन बॉन्ड है, और जी और सी के बीच है जो 3 हाइड्रोजन बॉन्ड है. फिर ये पॉलीन्यूक्लियर चेन दिया हुआ है. इधर 5' फॉस्फेट, और इधर पर दिखाया हुआ है 3' हाइड्रोक्साइल दिखाया हुआ है. 5' पर फॉस्फेट है और 3' पर है जो हाइड्रोक्साइल है. इसके बाद फिर आता है पैकेजिंग ऑफ द डीएनए हेलिक्स. टेकन द डिफरेंस बिटवीन द टू कंसेक्यूटिव बेस पेयर एज द 0.34 यानी कि 0.34 into 1 nanometer. If the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of the base pair with the difference between the two consecutive base pair, that is 6.6 into 9 base pair यानी 6.6 into 9 base pair into 0.34 into 9 base per base pair, it comes out to be approximately 2.2 meter. A length that is far greater than the dimension of a typical nucleus, approximately 10 to the minus 6 meter. How is such a long polymer packed in a cell? If the length of the E. coli DNA is 1.36 millimeter, can you calculate the number of the base pair in the E. coli? In the prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some protein that have the positive charge in a region termed as the nucleoid. The DNA in the nucleoid is organized in a large loop fed by the proteins. In eukaryotes, this organization is much more complex. There is a set of the positively charged basic proteins called the histone. A protein acquires charge depending upon the abundance of the amino acid residue with the charged fat chains. Histones are rich in the basic amino acid residue lysine and arginine. Both the amino acid residue carry positive charges in their fat chain. Histones are united to form a unit of the 8 molecule called the histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called a nucleosome. A typical nucleosome contains those who base pair of a DNA helix. Nucleosome constitutes the repeating unit of the structure in a nucleus called the chromatin. Thread-like stranded colored bodies can be seen in the nucleus. The nucleosome in the chromatin are seen as the beads on the string of structure when viewed under the electron microscope. Theoretically, how many beads nucleosome do you imagine are present in a mammalian cell? The beads on the string of structure in the chromatin is packaged to form the chromatin fiber that are further coiled and condensed at the metaphase stage of the cell division to form the chromosomes. The packaging of the chromatin at the higher level requires additional set of the proteins that collectively are referred to as non-histone chromosomal proteins (NHC). In a typical nucleus, some regions of the chromatin are loosely packed and often flat and are referred to as the euchromatin. The chromatin that is more densely packed and often dark are called as the heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. तो यहाँ पर यह न्यूक्लियोफॉर्म का पिक्चर दिया हुआ है जिसमें है कोर ऑफ द हिस्टोन मॉलिक्यूल डीएनए है, H1 हिस्टोन है, हिस्टोन ऑक्टामर है। फिर ये जो पिक्चर है इलेक्ट्रॉन माइक्रोस्कोप का पिक्चर है, बीड्स ऑन द स्ट्रिंग दिखाए हुए है। उसके बाद फिर आता है द सर्च फॉर द जेनेटिक मटेरियल। तो इवन दो द डिस्कवरी ऑफ द न्यूक्लियन बाय द मिसर एंड द proposition for the principle of the inheritance by the Mendel were almost at the same time, but that the DNA active genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by the Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and the numerous other scientists had narrowed the stretch to the chromosome located in the nucleus of most cells. But the question what molecule was actually the genetic material had not been answered. Transforming principle in 1928, Frederick Griffith, in a series of experiments with the Streptococcus pneumoniae, bacterium responsible for the pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism bacteria had changed in the physical form. When the Streptococcus pneumoniae pneumococcus bacteria are grown on a culture plate, some produce smooth shiny colonies while others produce the rough colonies. This is because the S-strain bacteria have a mucous polysaccharide coat while R-strain does not. Mice infected with the R-strain virulent die from the pneumonia infection, but mice infected with the R-strain do not develop the pneumonia. यानी कि S-strain को इंजेक्ट टू माइस किया तो माइस डाई हो गया, R-strain को इंजेक्ट टू माइस किया तो माइस अलाइव रहा. Griffith was able to kill the bacteria by hitting them. He observed that heat-killed S-strain bacteria injected into mice did not kill them. When he uh, when he injected a mixture of the heat-killed S and the and the S heat-killed S and the live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle transferred from the heat-killed S strain had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of the genetic material was not defined from his experiment. तो हीट किल्ड S-strain हीट किल को इंजेक्ट इनटू माइस में तो माइस लाइव रहा, लेकिन हीट S-strain हीट किल्ड और R-strain जो कि लाइव है इंजेक्ट इनटू माइस में तो माइस है जो डाई हो गया. फिर आता है बायोकेमिकल कैरेक्टराइजेशन ऑफ द ट्रांसफॉर्मिंग प्रिंसिपल. तो प्रायर टू द वर्क ऑफ द Oswald Avery, Colin MacLeod and the Maclean McCarthy, 1933 to 1944, the genetic material was thought to be protein. The worked to determine the biochemical nature of the transforming principle in the Griffith experiment. They purified the biochemicals protein, DNA, RNA, etc. from the heat-killed F cell to see which one could transform live R cells into the F cells. They discovered that DNA alone from this bacteria caused the R bacteria to become the transformed. They also discovered that protein digesting enzymes protease and the RNA digesting enzyme RNA did not affect transformation, so the transforming substance was not a protein or RNA. Digestion with the DNA did inhibit transformation, suggesting that the DNA caused the transformation. They could conclude that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between the DNA and the DNAs? उसके बाद फिर आता है द जेनेटिक मटेरियल इन द डीएनए द अनइक्विवोकल प्रूफ दैट डीएनए जेनेटिक मटेरियल केम फ्रॉम द एक्सपेरिमेंट ऑफ द अल्फ्रेड हर्शी एंड द मार्था चेज 1952 में द बर्केड विद द वायरस दैट अफेक्ट द बैक्टीरिया विद द बैक्टीरियोफाज द बैक्टीरियोफाज अटैच टू द बैक्टीरिया एंड इट्स जेनेटिक मटेरियल दैट enters the bacterial cell. The bacterial cell treats the viral genetic material as if it by its own and subsequently manufactures more biophage particles. Hershey and Chase worked to discover whether it was protein or DNA from the virus that entered the bacteria. They grew some virus on a medium that contained radioactive phosphorus and some other on a medium that contained radioactive sulfur. Biophage grown in the presence of the radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, virus grown on the radioactive sulfur contained radioactive protein but not radioactive DNA because DNA does not contain the sulfur. इसके बाद फिर आता है कि radioactive phages were allowed to attach E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with the virus that had radioactive DNA were radioactive, which indicating that DNA was the genetic material that passed from the virus to the bacteria. Bacteria that were infected with the virus had radioactive proteins were not radioactive. This indicates that the protein did not enter the bacteria from the virus. DNA is therefore the genetic material that is passed from the virus to the bacteria. तो ये बैक्टीरियोफाज हो गया, रेडियो एक्टिव F35 लेबल्ड प्रोटीन कैप्सूल हो गया, ये रेडियो एक्टिव फॉस्फोरस लेबल्ड डीएनए हो गया. No radioactive detected in the cells or radioactive F35 detected in the supernatant. ये इन्फेक्शन फिर ब्लेंडिंग और फिर सेंट्रीफ्यूगेशन करें. फिर रेडियोएक्टिव डिटेक्टेड इन द सेल्स, नो रेडियोएक्टिव डिटेक्टेड इन द सुपरनेटेंट. ये हर्फी चेज एक्सपेरिमेंट है. फिर आता है द प्रॉपर्टी ऑफ द जेनेटिक मटेरियल डीएनए वर्सेज आरएनए. For the foregoing discussion, it is clear that the debate between the proteins versus DNA as the genetic material was unequivocally resolved from the Hershey-Chase experiment. It became an established fact that it is the DNA that acts as the genetic material. However, it is subsequently become clear that in some viruses, RNA is genetic material. For example, tobacco, mosaic virus, COVID, bacteriophage, etc. Answer to some of the questions such as why DNA is the predominant genetic material, why the RNA performs dynamic function of the messenger and adapter had to be found from the difference between the chemical structure of the two nucleic acid molecules. Can you recall the two chemical difference between the DNA and the RNA? फिर आता है कि a molecule that can act as genetic material must fulfill the following criteria. पहला कि it should be able to generate its replication, it should be stable chemically and structurally, it should provide the scope for the slow changes mutation that are required for the evolution, or it should be able to express itself in the form of the Mendelian characters. If one examines each requirement one by one because of the rules of the base pairing and the complementarity, both the nucleic acid D and the RNA have the ability to direct their duplications. The other molecule in the living system such as the proteins fail to fulfill their first criteria itself. The genetic material should be stable enough not to change with the different stage of the life cycle, age, or with the change in the physiology of the organism. Stability is one of the property of the genetic material was very evident in the Griffith transforming principle itself that heat which killed the bacteria at least did not destroy some of the property of the genetic material. This can now easily be explained in the light of the DNA that the two strands of being complementary if separated by the heating some come together when appropriate conditions are provided. Further, two diphosphate group present at every nucleotide in the RNA is a reactive group and makes RNA liable and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to the RNA. Therefore, among the two nucleic acids, the DNA is better genetic material. In fact, the presence of the thymine at the place of the virus will also confer additional stability to the DNA. Detailed discussion about this require understanding of the process of the repair in the DNA, and we will study this process in the higher classes. Both DNA and RNA are able to mutate. In fact, RNA being the unstable mutate at a faster rate. Consequently, virus having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of the proteins and can easily express the character. RNA, however, is dependent on the RNA for the DNA, however, is dependent on RNA for the synthesis of the protein. The protein synthesizing machinery has evolved around the RNA. The above discussion indicate that both RNA and DNA can function as genetic material, but DNA being more stable is preferred for the storage of the genetic information. For the transmission of genetic information, RNA is the better. फिर आता है RNA world. That for foregoing discussion, an immediate question become evident, which is the first genetic material. It shall be discussed in a detail in a chapter on the chemical evolution, but briefly we shall highlight some of the facts and the points. RNA was the first genetic material. This is now enough evidence to suggest that essential life processes such as the metabolism, translation, splicing, etc. evolved around the RNA. RNA used to act as genetic material as well as the catalyst. There are some important biochemical reactions in the living system that are catalyzed by the RNA catalyst and not by the protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from the RNA with the chemical modification that make it more stable. DNA being double-stranded and having complementary strand further issues change by evolving a process of the repair. फिर आता है replication. So while proposing the double helical structure for the DNA, Bartman and the Crick had immediately proposed a scheme for the replication of the DNA to create the original statement that is as follows. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick 1953 में कहा था. The scheme suggested that the two strands could separate and act as a template for the synthesis of the new complementary strands. After the completion of the replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as the semi-conjugative DNA replication. तो यहाँ पर दिए हुए वाटसन क्रिक मॉडल फॉर दी सेमी कंजक्टिव डीएनए रेप्लिकेशन. उसके बाद फिर आता है द एक्सपेरिमेंटल प्रूफ कि इट इज नॉट प्रूवन दैट डीएनए रेप्लिकेट सेमी conservatively. It was found first in the E. coli and subsequently in higher organisms such as the plant and the human cells. Matthew Michelson and the Franklin Stahl performed the following experiment in 1958. They grew the E. coli in a medium containing NH4 fuel. N15 is the heavier isotope of the nitrogen as the only nitrogen source for many generations. The result by that N15 was incorporated into the newly synthesized DNA as well as the other nitrogen containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in the fusion chloride density gradient. Please note that N15 is a radioactive isotope and it can be separated from N14 only based on the density. फिर आता है कि then they transferred the cells into a medium with the normal NH14 fuel and took samples at various definite time intervals as the cells multiplied and extract the DNA that remained at the double-stranded helices. The various samples were separated independently on the fuel gradients to measure the density of the DNA. Can you recall Bart's centrifugal force and think why a molecule with higher molecule mass density would fragment fast? The result are shown in the figure 5.7. तो यहाँ पर दिए हुए सेपरेशन ऑफ द डीएनए बाय द सेंट्रीफ्यूगेशन, ग्रेविटेशनल फोर्स दिए हुए 20 मिनट, फिर उसके बाद 40 मिनट और में हाइब्रिड आया, फिर उसके बाद है जो लाइट बैंड और हाइब्रिड है जो आ गया. ये जनरेशन 1 है और ये है जो जनरेशन 2 है. तो यह है मिशेल्सन एंड द स्टॉहल का एक्सपेरिमेंट है. फिर Thus, the DNA was extracted from the culture one generation after the transfer from the N15 to N14 medium. That is, after 20 minutes, E. coli derived from the 20 minutes that had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is, after 40 minutes to second generation batch, composed of the equal amount of this hybrid DNA and of the light DNA. If E. coli was allowed to grow for the 80 minutes, then what would be the proportion of the light and the hybrid density DNA molecule? Very similar experiment involving use of the radioactive thymidine to detect the distribution of the newly synthesized DNA in the chromosome by preformed on the Vixia fava beans by the Taylor and the colleagues in 1958. The experiment proved that the DNA in the chromosome also replicates semi-conservatively. फिर आता है the machinery and the enzyme. In the living cells such as E. coli, the process of the replication requires a set of the catalyst enzymes. The main enzyme is referred to as the DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of the deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of the nucleotides in a very short of time. E. coli that has only 4.6 units in the power 6 base pair compared it with the human whose diploid content is 6.6 into 10 to the power 9 base pair completes the process of the replication within the 18 minutes. That means the average rate of the polymerization had to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with very high degree of the accuracy. Any mistake during the replication would result into the mutations. Furthermore, energetically replication is a very expensive process. Deoxyribonucleoside triphosphate serves dual purpose. In addition to acting as a substrate, they provide energy for the polymerization reaction. The two-terminal phosphate in a deoxynucleoside triphosphate are high-energy phosphates, same as in the case of the ATP. In addition to the DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of the replication with the high degree of the accuracy. For the long DNA molecules, since the two strands of the DNA cannot be separated in its entire length due to high energy requirement, the replication occurs with a small opening of the DNA helix referred to as the replication fork. The DNA-dependent DNA polymerase catalyze the polymerization only in the one direction, that is 5' to 3'. This creates some additional complication at the replicating fork. Consequently, on one strand a template with the polarity 3' to 5', the replication is continuous, while on the other the template with the polarity 5' to 3', it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase. The DNA polymerase on their own cannot initiate the process of the replication. Also, the replication does not initiate randomly at any place in the DNA. There is a definite region in the E. coli DNA where the replication originates. Such regions are termed as the origin of the replication. It is because of the requirement of the origin of the replication that a piece of the DNA is needed to be propagated during the recombinant DNA procedures requires a vector. The vector provides the origin of the replication. Further, not every detail of the replication is understood well. In eukaryotes, the replication of the DNA takes place at the S phase of the cell cycle. The replication of the DNA and the cell division cycles should be highly coordinated. A failure in the cell division after DNA replication results into the polyploidy, a chromosomal anomaly. We will learn the detailed nature of the origin and the process occurring at this site in the higher classes. तो ये दिया हुआ है रिप्लिकेटिंग फॉर्क दिया हुआ है न्यूली सिंथेसाइज स्ट्रैंड्स हो गया ये कंटिन्यूस सिंथेसिस हो गया ये टेम्पलेट पेरेंटल स्ट्रैंड्स हो गया और इधर हो गया डिसकंटिन्यूस सिंथेसिस हो गया उसके बाद फिर आता है ट्रांसक्रिप्शन द प्रोसेस ऑफ कॉपिंग जेनेटिक इन्फॉर्मेशन फ्रॉम द वन स्ट्रैंड ऑफ द DNA into RNA is termed as the transcription. Here also the principle of the complementarity governs the process of the transcription, except the adenosine complements now forms base pair with the uracil instead of the thymine. However, unlike in the process of the replication, which one fits in the total DNA of an organism gets duplicated, in transcription only a segment of the DNA and only one of the strands is copied into the RNA. This necessitates defining the boundaries that would demarcate the region and the strand of the DNA that would be transcribed. Why both the strands are not copied during the transcription has the simple answer. First, in both strands act as a template. They would code for the RNA molecules, but different sequence. Remember, complementarity does not mean identical, and in turn, if they code for the proteins, the sequence of the amino acid in the protein would be different. Hence, the one segment of the DNA would be coding for the two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules produced simultaneously would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of the transcription would become a futile one. फिर आता है ट्रांसक्रिप्शन यूनिट अ ट्रांसक्रिप्शन यूनिट इन द डीएनए इज डिफाइंड प्राइमरली बाय द थ्री जीन्स इन द डीएनए पहला है प्रोमोटर द स्ट्रक्चरल जीन और फिर आता है टर्मिनेटर तो there is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that if 5' to 8' the strand that has the polarity of 3' to 5' act as a template and is also referred to as the template of strand. The other strand which has the polarity 5' to 8 3' and the sequence is same as the RNA except thymine at the place of the uracil is displaced during the transcription. Strangely, this strand which does not code for anything is referred to as the coding strand. All the reference point while defining a transcription unit is made with the coding strand. To explain this, the point or hypothetical sequence from transcription unit is रिप्रेजेंटेड बिलो। तो 3-5 हो गया, टेम्पलेट ऑफ स्टैंड हो गया, और इधर ये कोडिंग ऑफ स्टैंड हो गया। कैन यू राइट द सीक्वेंस ऑफ द आरएनए ट्रांसफर्ड फ्रॉम द एबव डीएनए? तो ये हो गया सीमेटिक स्ट्रक्चर ऑफ द ट्रांसक्रिप्शन यूनिट। ये ट्रांसक्रिप्शन ऑफ स्टार्ट फाइट है, प्रमोटर हो गया, स्ट्रक्चरल जीन हो गया, टेम्पलेट ऑफ स्टैंड हो गया, कोडिंग ऑफ स्टैंड हो गया, इधर हो गया टर्मिनेटर। तो कहता है कि द प्रोमोटर एंड द टर्मिनेटर फ्लैंक्स द स्ट्रक्चरल जीन इन द ट्रांसक्रिप्शन यूनिट। द प्रोमोटर इज said to be located towards the 5' upstream of the structural gene. The reference we made with respect to the polarity of the coding strand. It is a DNA sequence that provides for binding fat for a RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and the coding of strands. By switching its position with the terminator, the definition of the coding and the template of strands could be reversed. The terminator is located towards the 3' downstream of the coding strand, and it is usually defines the end of the process of the transcription. There are additional regulatory sequences that may be present further upstream or the downstream to the promoter. Some of the properties of this sequence shall be discussed while dealing with the regulation of the gene expression. इसके बाद फिर आता है ट्रांसक्रिप्शन यूनिट एंड द जीन। तो ए जीन इज डिफाइंड एज द फंक्शनल यूनिट ऑफ द इनहेरिटेंस, दो देयर इज नो एम्बिग्यूटी दैट द जीन्स आर लोकेटेड इन डीएनए, इट इज डिफिकल्ट टू लिटरली define a range gene in the terms of the DNA sequence. The DNA sequence coding for the tRNA or the RNA molecule also defines a gene. However, by defining a histone as a segment of the RNA coding for a polynucleotide, polypeptide, the structural gene in a transcription unit could be said as the monohistonic, mostly in the eukaryotes, or polyhistonic, mostly in the bacteria or the prokaryotes. In the eukaryotes, the monohistonic structural genes have interrupted coding sequence. The genes in the eukaryotes are split. The coding sequence or expressed sequence are defined as exons. Exons are said to be those sequences that appear in the mature or processed RNA. The exons are interrupted by the introns. Introns for intervening sequences do not appear in the mature or the processed RNA. The split gene arrangement further complicates the definition of a gene in the terms of the DNA segment. Inheritance of a character is also affected by the promoter and the regulatory sequence of a structural gene, and sometimes the regulatory sequences are loosely defined as the regulatory genes, even though these sequences do not code for any RNA or the protein. So at that types of the RNA and the process of the transcription. So in the bacteria, there are three major types of the RNA: mRNA, messenger RNA, tRNA, transfer RNA, and the rRNA, ribosomal RNA. All three RNA are needed to synthesize a protein in the cell. The mRNA provides the template, tRNA brings the amino acids and its the genetic code, and the rRNA play a structural and the catalytic role during the translation. There is single DNA-dependent RNA polymerase that catalyzes the transcription of all the types of the RNA in a bacteria. RNA polymerase binds to the promoter and initiates the transcription initiation. It uses nucleoside triphosphate as the substrate and polymerizes in a template-dependent fashion following the rule of the complementarity. It somehow also facilitates the opening of the helix and continuous elongation. Only a short stretch of the RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off so as the RNA polymerase. This results in the termination of the transcription. तो दिए हुए द प्रोसेस ऑफ द ट्रांसक्रिप्शन इन द बैक्टीरिया, इनिशिएशन हो गया, फिर इलोंगेशन हो गया, फिर आता है टर्मिनेशन। तो इनिशिएशन में आरएनए पॉलीमरेज सिग्मा फैक्टर आकर लग गया, और इस इलोंगेशन में है जो इलोंगेट हो रहा है, और फिर टर्मिनेशन में है जो रो फैक्टर आके है जो लग जाता है। इसके बाद इंसर्ट कहता है कि एन इंट्रिगिंग क्वेश्चन इज दैट हाउ इज द आरएनए पॉलीमरेज एबल टू कैटलाइज ऑल द थ्री स्टेप्स विच आर इनिशिएशन, इलोंगेशन एंड टर्मिनेशन. The RNA polymerase only capable of catalyzing the process of the elongation. It associates transiently with the initiation factor sigma and the termination factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and the translation take place in the same compartment, there is no separation of the cytosol and nucleus in the bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in the bacteria. In eukaryotes, there are two additional complexities. There are at least three RNA polymerase in the nucleus in addition to the RNA polymerase found in organelles. There is a clear-cut division of the label. The RNA polymerase will transcribe rRNAs uh, 28F, 18F, and 5.8F by the RNA polymerase III is responsible for the transcription of the tRNA, 5F RNA, and FNRNA, small nuclear RNA. The RNA polymerase II transcribe precursor of the mRNA, the heterogeneous nuclear RNA, hnRNA. The second complexity is that primary transcripts contain both the exons and the introns and are the functional, non-functional, hence it is subjected to a process called the splicing, where the introns are removed and the exons are joined in a defined order. HNRA undergoes additional processing called the capping and the tailing. In the capping, an unusual nucleotide, methylguanosine triphosphate, is added to the 5' of the hnRNA. In the tailing, adenylyl residue, those sequence of bases are added at the 3' template independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for the translation. The significance of such complexity is now beginning to be understood. The split gene arrangements represents probably an ancient feature of the genome. The presence of the introns is reminiscent of the antiquity, and the process of splicing represents the dominance of the RNA world. In recent times, the understanding of the RNA and the RNA-dependent process in the living system has assumed more importance. तो ये प्रोसेस ऑफ द ट्रांसक्रिप्शन यूकैरियोट्स दिखा रहा है. इसमें जो है कैपिंग किए 5' पर, फिर 3' पर है ये पॉली ए टेल से पॉली एडेनालाइजेशन किए और ये मैसेंजर आरएनए जो बन गया, जिसमें है जो हम इंट्रॉन्स को हटा दिए हैं और सिर्फ एक जॉन्स इसमें है. इसके बाद पर आता है जेनेटिक कोड. तो डरिंग दी इन्फेटिक कहता है कि during the replication and the transcription, a nucleic acid was copied to form the another nucleic acid. Hence, these processes are easy to conceptualize on the basis of the complementarity. The process of the translation requires transfer of the genetic information from a polymer of the nucleotides to synthesize a polymer of the amino acids. Neither does any complementarity exist between nucleotides and the amino acid, nor could any be drawn theoretically. There existed ample evidence, though, to support the notion that change in the nucleic acid genetic material were responsible for the change in the amino acids in the proteins. This led to the proposition of a genetic code that could direct the sequence of the amino acid during the synthesis of the protein. If determining the biochemical nature of the genetic material and the structure of the DNA was very exciting, the proposition and the deciphering of the genetic code were almost challenging. In very true sense, it required involvement of the scientists from the several disciplines: physicists, organic chemists, biochemist, and the geneticists. It was the George Gamow, a physicist, who argued that since there are only 4 bases and if they have to code for the 20 amino acids, the code should constitute a combination of the bases. He suggested that in order to code for all the 20 amino acids, the code should be made of the 3 nucleotides. This was a very bold proposition because a permutation combination of the 4k power q would generate 54 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by the Hargun Khwana was instrumental in synthesizing the RNA molecule with the defined combination of the bases, homopolymers, and the copolymers. Marshall-Nirenberg cell-free system for the protein synthesis finally helped the code to be deciphered. Several Febro-Okwar enzymes, polynucleotide phosphorylase, were also helpful in polymerizing RNA with defined sequence in template-independent manner, enzymatic synthesizing of the RNA. Finally, a checkerboard for the genetic code by prepared, which is given in table 5.1. The codons for the various amino acid, ये फर्स्ट प्रोपोजिशन इधर है, सेकंड प्रोपोजिशन इधर है, थर्ड पोजिशन दिए हुए. तो द साइलेंट फीचर ऑफ द जेनेटिक कोड आर एज फॉलोज कि the codon is triplet. 51 codons code for the amino acids and 3 codons do not code for any amino acid since they function as the stop codons. Some amino acids are coded by more than one codon, hence the code is degenerate. फिर the code is read in the mRNA in a contiguous fashion. There are no punctuations. उसके बाद आता है कि the code is nearly universal. For example, from bacteria to the human, UUU would code for the phenylalanine. Some exceptions to this rule have been found in the mitochondrial codons and some protozoans. AUG had a dual function which codes for the methionine and it also acted as the initiator codon. UAA, UAG, and the UG are the stop terminator codons. If following is the sequence of nucleotides in the mRNA, predict the sequence of the amino acid coded by it, take help of the checkerboard. तो Now try in the opposite. Following is the sequence of the amino coded by the mRNA. Predict the nucleotide in the mRNA. Do you face any difficulty in predicting the opposite? Can you now correlate with the two properties of genetic code you have learned? Plus, the mutations and genetic code. The relationship between the genes and the DNA are best understood by the mutations of studies. You have studied about the mutation and its effect in the chapter 4. Effects of the large deletions and the rearrangements in the fragment of the DNA are easy to comprehend. It may result in a loss or gain of a gene or for a function. The effect of the point mutations will be explained here. A classical example of the point mutation is a change of the single base pair in the gene of for a beta globin chain that results in the change of the amino acid residue glutamate to valine. It results into a disease condition called the sickle cell anemia. Effect of the point mutation that inserts or deletes a base in the structural gene can be better understood by the following simple example. Consider a statement that is made up of the following words, each have the three letters of genetic code. If we insert a letter B in between the hash and the red and rearrange the statement, it would read as the follow. Similarly, if we read insert two letters in the same place, say BI, it would read. Now if we insert three letters together, say big, the statement would read as this one. The same exercise can be repeated by deleting the letters R, E and D one by one and rearranging the statement to make a triplet word. The conclusion by from the above is obvious. Insertion or deletion of one or two bases changes the reading frame from the point of the insertion or deletion. However, such mutations are referred to as the frame shift insertion or the deletion mutation. Insertion or deletion of the three or its multiple bases insert or delete in one or multiple codon. Hence, one or multiple amino acids on the reading frame remains unaltered from the that point onwards. इसके बाद फिर आता है tRNA, the adaptive molecule. From the very beginning of the proposition of the code, it was clear to the Francis Crick that there has to be a mechanism to read the code and also to link it with the to the amino acid because amino acid have no structural speciality to read the code uniquely. He postulated the presence of an adapter molecule that would, on one hand, read the code and on the other hand would bind to the specific amino acid. The tRNA, then called the snRNA soluble RNA, by known before the genetic code was postulated. However, its role as an adapter molecule was refined much later. tRNA has an anticodon loop that has bases complementary to the code and it also has an amino acid receptor end to which it binds to the amino acid. tRNA are specific for each amino acid. For the insertion there is another specific tRNA that is referred to as the inserted tRNA. There are no tRNA for the stop codons. In the figure 5.12, the secondary structure of the tRNA has been depicted that looks like a clover leaf. In actual, the structure of the tRNA is a compact molecule which looks like the inverted L. तो ये यहाँ पर दिया हुआ है एंटीकोडॉन दिया हुआ है, यहाँ पर mRNA दिया हुआ है, ये tRNA एडेप्टर मॉलिक्यूल दिए हुए। फिर उसके बाद आता है ट्रांसलेशन। The translation refers to the process of the polymerization of the amino acid to the form of polypeptide. The order and the sequence of the amino acid are defined by the sequence of the bases in the mRNA. The amino acids are joined by a bond which is known as the peptide bond. Formation of a peptide bond requires the energy. Therefore, in the first phase itself, amino acids are activated in the presence of the ATP and linked to the cognate tRNA process commonly called the charging of the tRNA or the amino acylation of the tRNA to be more specific. If two such tRNA are brought close enough, the formation of the peptide bond between them would be favored energetically. The presence of the catalyst would enhance the rate of the peptide bond formation. The cellular factory responsible for the synthesizing protein in the ribosome. The ribosome consists of the structural RNA and about 80 different proteins. In its active state, it is used as the two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of the translation of the mRNA to the protein begins. There are two sites in the large subunit, and the subsequent amino acid to bind and thus are close enough to each other for the formation of the peptide bond. The ribosome also acts as a catalyst 23-sRNA in the bacteria in the enzyme ribosome for the formation of the peptide bond. A translational unit in the mRNA sequence of the RNA that is flanked by the start codon AOG and the stop codon encodes for a polypeptide. And mRNA also has some additional sequences that are not translated and are referred to as the untranslated regions UTR. The UTR are present at both the 5' before the start codon and the 3' after the stop codon. They are required for the efficient translation process. For the initiation, the ribosome binds to the mRNA at the start codon AOG that is recognized only by the initiated tRNA. The ribosome proceeds to the elongation phase of the protein synthesis. During this phase, After the complex compound of an amino acid linked to the tRNA sequentially bind to the appropriate code in the mRNA by forming complementary base pair with the tRNA anticodon. The ribosome moves from the codon to the codon along the mRNA. Amino acids are added one by one, translated into the polypeptide sequence detected by the DNA and imprinted by the mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. तो ये यहाँ पर ट्रांसलेशन दिखा रहा है यहाँ पर. उसके बाद फिर आता है regulation of the gene expression. So regulation of the gene expression refers to a very broad term that may occur at the various levels. Considering that the gene expression results in the formation of a polypeptide, it can be regulated at the several levels. In the eukaryotes, the regulation could be exerted at transcriptional level, formation of primary transcript, processing level, regulation of the splicing, transport of the mRNA from nucleus to the cytoplasm, and the translational level. तो इसमें आता है कि the genes in a cell are expressed to perform a particular function or a set of the function. For example, if an enzyme called beta-galactosidase is synthesized by the E. coli, it is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. The bacteria use them as a source of the energy. Hence, if the bacteria do not have lactose around them to be utilized for the energy source, they would no longer require the synthesis of the enzyme beta-galactosidase. Therefore, in the simple terms, if it is the metabolic, physiological, environmental condition that regulate the expression of the gene, the development and the differentiation of the embryo into adult organism are also a result of the coordinated regulation of the expression of the several sets of the genes. In prokaryotes, the control of the rate of the transcriptional limit initiation is the predominant site for the control of the gene expression in a transcriptional unit. The activity of the RNA polymerase at a given promoter is in turn regulated by the interaction with the accessory proteins, which affects its ability to recognize of start sites. These regulatory proteins can act both positively activators and negatively repressor. The accessibility of the promoter regions of the prokaryotic DNA is in many cases regulated by the interaction of the proteins with the sequence terminated by the operators. The operator regions with addition to the promoter elements in the most of the operons, and in most cases the sequence of the operator bind a repressor protein. Each operon has its specific operator and a specific repressor. For example, lag operator is present only in the lag operon and it interacts specifically with the lag repressor only. फिर आता है lag operon. So the elucidation of the lag operon by alpha result of the close association between the genetist Francois Jacob and the biochemist Jacques Monod. They bear the first elucidated transcriptionally regulated system in the lag operon. Here lag refers to the lactose. A polycystonic structural gene is regulated by a common promoter and a regulated gene. Such arrangements is very common in bacteria and is referred to as the operon. To name few such examples, lag operon, tryptophan-operon, rabinoidopteron, histopteron, valine-operon, etc. The lag operon consists of the one regulatory gene, the I gene. Here the term I does not refer to the inducer, rather it is derived from the word inhibitor. And the three structural genes, gene Z, Y, and A. The I gene codes for the repressor of the lag operon. The Z gene code for the beta-galactosidase, beta-gal, which is primarily responsible for the hydrolysis of the disaccharide lactose into its small molecular unit galactose and the glucose. The Y gene code for the permease, which increases permeability of the cell to the beta-galactosidase. The A gene encodes for transacetylase. Hence, all the three gene products in the lag operon are required for the metabolism of the lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. ये द लैक ऑपरॉन दिए हुए ये repressor mRNA है ये repressor हो गया repressor bind to the operator region and prevents the RNA polymerase from transcribing the operon. ये repression mRNA हो गया ये inducer effect किया बीटा galactosidase permease और transacetylase ZYA GPT से है जो बन रहा है. इसके बाद फिर आता है कि lactose is the substrate for the enzyme beta-galactosidase and it regulates switching on and off of the operon, hence it is determined as the inducer. In the absence of the preferred carbon source such as the glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of the permease. Remember, every low level of the expression of the lag operon has to be present in the cell all the time, otherwise lactose cannot enter the cell. The lactose then initiates the operon in the following manner. The repressor of the operon is synthesized at all the time constructively from the I zone. The repressor protein bind to the operator region of the operons and prevent RNA polymerase from transcribing the operon. In the presence of a new repressor such as lactose or the allolactose, the repressor is inactivated by the interaction with the inducer. This allows the RNA polymerase access to the promoter and the transcription proceeds. Essentially, regulation of the lag operon can also be visualized as a regulation of the enzyme synthesized by its substrate. Remember, glucose or the galactose cannot act as the inducer for the lag operon. Can you think how long the lag operon would be expressed in the presence of the lactose? Regulation of the lag operon by the repressor is referred to as the negative regulation. Lag operon is under the control of the positive regulation as well, but it is beyond the scope of the discussion at this level. फिर इसके बाद ह्यूमन जीनोम प्रोजेक्ट के बारे में इंसर्ट बता रहा है कि in the preceding section we have learned that it is the sequence of the bases in the DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequence. If two individuals differ, then their DNA sequence would also be different, at least at some places. These assumptions led to the quest of the finding out the complete DNA sequence of the human genome with the establishment of the genetic engineering technique where it was possible to isolate and clone any piece of the DNA and ability of the simple and the fast technique for determining DNA sequences. A very ambitious project of the sequencing human genome was launched in 1990. Human Genome Project was called a mega project. You can imagine that the magnitude and the requirements for the project if we simply define the aims of the project as follows. Human genome is said to have approximately 3 into 10 to the 9 base pair, and if the cost of sequencing required is $3 per base, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion. Further, if the obtained frequencies were to be stored in a typed form in the books, and each page of the book contained 1,000 letters, and each book contained 1,000 pages. The 30 such books would be required to store the information of the DNA sequence from a single human cells. The enormous amount of the data expected to be generated also necessitated the use of the high-speed computational devices for the data storage and retrieval and analysis. HGP was closely associated with the rapid development of a new area in the biology called the bioinformatics. फिर आता है goals of the HGP. तो some of the important goals of the HGP are as follows: identify all the approximately 20,000 to 25,000 genes in the human DNA, determine the sequence of the 3 billion chemical bases pair that make up the human DNA, store this information in a database, improve the tools for the data analysis, transfer related technology to the other sectors such as industries, address the ethical, legal, and the social issues that may arise from the project. The Human Genome Project by a 13-year project coordinated by the US Department of Energy and the National Institute of Health. During the early years of the HGP, the Wellcome Trust UK became a major partner. Additional contributions come from the Japan, France, Germany, China, and others. The project was completed in 2003. Knowledge about the effect of the DNA variation among individuals can lead to the revolutionary new ways to diagnose, treat, and someday prevent the thousands of the disorder that affect the human beings. Besides providing clue to the understanding human biology, learning about the non-human organism DNA sequence can lead to an understanding of the natural capability that can be applied towards solving challenges in the healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as the bacteria, yeast, Phenorabiditis elegans, a free-living non-pathogenic nematode, Drosophila plants, Rhabdopsis have also been sequenced. Methodology: the methods involve two major approaches. One approach focused on identifying all the genes that are expressed by the RNA, referred to as the expressed gene sequence tags. The other took the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequence, and later assigning different regions in the sequence which function a term referred to as the sequence annotation. For sequencing the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall DNA is a long polymer, and there are technical limitations in sequencing very long pieces of the DNA and cloned in a suitable host using specialized vectors. The cloning resulted into amplification of each piece of the DNA fragment so that it subsequently could be sequenced with the ease. The commonly used host were bacteria and the yeast, and the vectors were called as the BAC, bacterial artificial chromosome, and the YAC, yeast artificial chromosome. The fragments were sequenced using the automated DNA sequencer that worked on the principle of the method developed by the Frederick Sanger. Remember, Sanger is also created by developing method for the determination of the amino acid sequence in the proteins. These sequences are then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences by humanly not possible. Therefore, a few slight computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in the May 2006. This was the last of the 24 human chromosome, 22 autosomes of the X and Y to be sequenced. Another challenging task was assigning the genetic and the physical map from the genome. This was generated using the information on the polymorphism of the restriction in the nuclear site and some repetitive DNA sequence known as the microsatellite. One of the applications of the polymorphism in the repetitive DNA sequence will be explained in the next section of the DNA finger printing. तो ये दिया हुआ है रिप्रेजेंटेटिव डायग्राम ऑफ द ह्यूमन जीनोम प्रोजेक्ट है जो दिए हुए हैं. इसके बाद फिर आता है साइलेंट फीचर्स ऑफ द ह्यूमन जीनोम प्रोजेक्ट. तो सम ऑफ द साइलेंट फीचर्स from the silent observation drawn from the human genome project are as follow. The human genome contains 3164.5 million base pair. The average gene consists of the 3000 base pair, but size vary greatly, with the largest known base human gene being dystrophin at 2.4 million base pair. The total number of the genes is estimated at this are much lower than the previous estimate of 80,000 to 1,40,000 genes. Almost 99% of the nucleotide base is actually the same in all the people. इसके बाद कहता है कि द फंक्शन आर अननोन फॉर ओवर 50% ऑफ द डिस्कवर्ड जीन्स, लेस देन 2% ऑफ द जीनोम्स कोड फॉर द प्रोटीन. Repeated sequences make up a very large portion of the human genome. Repetitive sequences are stretches of the DNA sequence that repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding function, but they shed light on the chromosome structure, dynamics, and the evolution. Chromosome 1 has the most genes, 2968, and the Y has the fewest, 231. Scientists have identified about 1.4 million locations where single base DNA differentiation (SNP) single nucleotide polymorphism pronounced as the SNP occur in the human beings. This information permits to revolutionize the process of finding chromosomal location for the disease-associated sequence and tracing human history. फिर आता है application and the future challenges. Deriving the meaningful knowledge from the DNA sequence will define the research through the coming decades, leading to an understanding of the biological system. These must task will require the expertise and the creativity of the tens of the thousands of the scientists from the varied disciplines in both the public and the private sectors worldwide. One of the greatest impacts of having the HG sequence may will be enabling to a readily new approach to biological research. In the past, researchers studied one or few genes at a time. With the whole genome sequence and a new high through output technology, we can approach questions systematically and on a much broader scale. They can study all the genes in the genome, for example, all the transcript in a particular tissue or organ or a tumor, or how tens of the thousands of the genes and the proteins work together in interconnected networks to orchestrate the chemistry of the life. इसके बाद फिर आता है DNA fingerprinting. तो as stated in the preceding section, 99.9% of the base sequence among human is the same. Assuming human genome at 3 into 10 to the 9 base pair, in how many bases sequence would there be differences? It is these differences in the sequence of a DNA which make every individual unique in their phenotypic appearance. If one aims to find out genetic differences between the two individuals or among the individuals of a population, sequencing the DNA every time would be a daunting and expensive task. Imagine trying to compare two sets of 10 to 10 to the 6 base pair. DNA fingerprinting is a very quick way to compare the DNA sequence of any two individuals. DNA fingerprinting involves identifying differences in some specific regions in the DNA sequence called the repetitive DNA, because in this sequence a small stretch of DNA is repeated many times. These repeated DNA are separated from the bulk genomic DNA at a different peak during the density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as the satellite DNA. Depending upon each composition, a T-rich or GC-rich length of the segment and the number of the repeated units, the satellite DNA is classified into many categories, such as the microsatellite, mini-satellites, etc. These sequences normally do not code for any proteins, but they form a large portion of the human genome. These sequences show a high degree of the polymorphism and form the basis of the DNA fingerprinting. Since DNA form every tissue such as the blood, hair follicle, skin, bones, liver, sperm, etc. perform from an individual for the same degree of the polymorphism, they become very useful identification tools in the forensic application. Further, as the polymorphism are inheritable from the parents to the children, DNA fingerprinting is the basis of the paternity testing in the case of the disputes. फिर कहता है कि the polymorphism in the DNA sequence is the basis of the genetic mapping of the human genome as well as the DNA fingerprinting. It is essential that we understand what DNA polymorphism means in the simple terms. Polymorphism variations at the genetic level arise due to the mutations. Recall different kinds of the mutation and the effect that we have already studied in the chapter 4 and in the preceding section in this chapter. New mutations may arise in an individual either in the somatic cells or in the germ cells, cells that generate gametes in the sexually reproducing organism. If a germ cell mutation does not seriously impair individual's ability to have offspring who can transmit the mutation, it can spread to the other members of the population through the sexual reproduction. Allelic again recall the definition of the allele from the chapter 4. Sequence variation has traditionally been described as a DNA polymorphism. If more than one variant allele at a locus occurs in a human population with a frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it refers to as a DNA polymorphism. The probability of such variation to be observed is in the non-coding DNA sequence would be higher as mutation in these sequences may not have any immediate effect impacting an individual's reproductive ability. These mutations keep on accumulating generations after generation and form one of the basis of the variability of polymorphism. There is a variety of the different types of the polymorphism ranging from the single nucleotide chain to very large scale changes. For evolution and speciation, such polymorphism may play important role and we will study these in detail at higher classes. The technique of the DNA fingerprinting was initially developed by the Alex Jeffrey. He used the DNA as a probe that show very high degree of the polymorphism. These were called as the variable number of the tandem repeats (VNTR). The technique as used earlier involved the southern blot hybridization using the radio-labeled VNTR as a probe. It included isolation of the DNA, titration of the DNA by the restriction endonuclease, separation of the DNA fragments by the electrophoresis, transferring blotting of the separate DNA fragments to the synthetic membranes such as the nitrosilage or nylon, hybridization using the labeled VNTR probe, and detection of the hybrid DNA fragments by autoradiography. A schematic representation of the DNA fragment is shown in figure 5.16. So the VNTR belongs to a class of the satellite DNA referred to as the mini satellite, a small DNA sequence arranged randomly in many copy numbers. The copy numbers vary from the chromosome to nucleome in an individual. The numbers of the repeats show very high degree of the polymorphism. As a result, the size of the VNTR vary in the size from the 0.1 to 20 kb. Consequently, after hybridization, the VNTR probe, the autoradiogram gives many bands of the different sizes. These bands give a characteristic pattern for individual DNA. It refers from the DNA individual to individual in a population, except in the case of the monozygotic identical twins. The sensitivity of the techniques has been increased by the use of the polymerase chain reaction (PCR). We will study consequently DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to the application in the forensic sciences, it has much wider applications such as in determining the population and genetic diversity. Currently, many different probes are used to generate the DNA fingerprints. तो ये दिया हुआ इधर schematic representation of the DNA fingerprinting. Fewer representative chromosomes have been found to contain different copies of the VNTR. For the sake of understanding, different color schemes have been used to trace the origin of each band in the gel. The two alleles, paternal and the maternal, of a chromosome also contain different copies of the VNTR. It is clear that the binding pattern of the DNA from the CAM scheme match individual B and not with the A. तो पैटर्नल मैटर्नल डीएनए फ्रॉम द ए, डीएनए फ्रॉम द बी, और इधर है जो डीएनए फ्रॉम द क्राइम सी. एम्पलीफाइड डीएनए रिपीट्स बाय द साइज ऑन अ जेल गिव्स अ डीएनए फिंगर प्रिंट. तो इसका समरी है कि न्यूक्लिक एसिड आर लॉन्ग पॉलीमर्स ऑफ द न्यूक्लियोटाइड्स while DNA stores the genetic information. RNA mostly helps in the transfer and the expression of the information, though DNA and RNA both function as genetic material, but DNA being chemically and structurally more stable is better genetic material. However, RNA is the first to evolve and DNA by derivation from the RNA. The hallmark of the double-stranded helical structure of the DNA is the hydrogen bonding between the bases from the opposite strand. The rule is the adenine pairs with the thymine through the 2H bonds and the guanine with the cytosine through the 3H bonds. This makes one strand complementary to the other. The DNA replicates semi-conservatively. The process is guided by the complementary H bonding. A segment of DNA that codes for the RNA, maybe in the simplistic term, can be referred to as the gene. DNA transcription, also one of the strands of the DNA, acts as a template to direct the synthesis of the complementary RNA. In bacteria, the transcribed mRNA is functional, hence can directly be translated In eukaryotes, the gene is split. The coding sequence exons are interpreted by the non-coding sequence introns. Introns are removed and exons are joined to produce the functional RNA by splicing. The messenger RNA contains the base sequence that are read in a combination of the three to make the triplet genetic code to code for an amino acid. Genetic code is read again on the principle of the complementarity by the tRNA that act as the adapter molecule. There are specific tRNA for every amino acid. The tRNA binds to the specific amino acid at one end and pairs through the H bonding with the code on the mRNA through its anticodons. The site of the transcription protein synthesis is riboform, which bind to the mRNA and provide platform for joining of the amino acid. One of the rRNA acts on a catalyst for the peptide 1 formation, which is an example of the RNA enzyme, ribozyme. Translation is a process that has evolved around the RNA, indicating that life began around the RNA. Since transcription and translation are energetically very expensive process, these have to be tightly regulated. Regulation of the transcription is the primary step for the regulation of the gene expression. In bacteria, more than one gene is arranged together and regulated in the units called as the operon. Lac operon is the prototype operon in the bacteria which codes for the gene responsible for the metabolism of the lactose. The operon is regulated by the amount of the lactose in the medium where the bacteria are grown. Therefore, this regulation can also be viewed as the regulation of the enzyme synthesized by the substrate. The Human Genome Project was a mega project that aimed to sequence every base in the human genome. The project has yielded much new information, many new areas and avenues have opened up as a consequence of the project. DNA fingerprinting is a technique to find out variations in the individuals of a population at a DNA level. It works on the principle of the polymorphism in the DNA sequence. It has immense application in the field of the forensic science, genetic biodiversity, and the evolutionary biology.